Okay, so the last 30 seconds of that video told you that the transforming principle is DNA. So, again, Avery, McLeod, and McCarty, 1944 publication on the nature of the transforming principle was the first direct experimental proof that it was DNA and not proteins. The studies, just like the video, Griffith used the mice and pneumonia, a virulent and non-virulent strain of pneumonia, and one killed and the other one didn't. And then that laid the foundation for Avery, McLeod, and McCarty. Also, same thing. He used the polysaccharide capsule, and which was the compound required for the capsule synthesis. Avery, McLeod, and McCarty published their results in 1944 and reportedly obtaining the transforming principle and demonstrated that the transforming principle was in fact DNA and not protein. But I mean, I feel like there were still doubters here. But so here is again just a picture of the video that you just saw. Now let's talk about these guys. This is Hershey and Chase. Now, I think they're adorable, but you should know that he continued to publish after this without her name on it, even though she did the research. So here's the experiment that they
In 1953, Alfred Hershey and Martha Chase
confirmed DNA's preeminent role in genetics by demonstrating that DNA is the genetic material of a virus called phage T2. The phage, which infects E. coli, consists of a head, sheath, tail, and base plate made of different proteins. DNA is packaged within the head of the virus. When T2 comes in contact with E. coli, the phage attaches to the bacterium by its tail. Next, the phage injects genetic material into the cell. The genetic material directs bacterial enzymes to produce viral offspring. When the life cycle is complete, 100 to 200 progeny phages have been assembled inside each bacterium. The bacterium breaks open, or lyses, and the phages are released. Since T2 consists of only DNA and protein, Hershey and Chase reasoned that the genetic material must be one of the two components, so they designed an experiment to determine which it is. For their experiment, the scientists prepared T2 phages that either had radioactive DNA or radioactive proteins. To make the components radioactive, Hershey and Chase infected E. coli with T2 and grew the bacterial cells in two different culture media, one containing the radioactive isotope phosphorus-32 and the other containing the radioactive isotope sulfur-35. All the phages released from the bacteria grown in the phosphorus-32 medium had radioactively labeled DNA, while all the phages released from the bacteria grown in the sulfur-35 medium had radioactively labeled protein.
Okay, so they radio-labeled the DNA, and they radio-labeled the protein, and now we wait to see which one actually infects the cell.
In the next phase of their experiment, Hershey and Chase infected E. coli with the two types of labeled T2. They then used a blender and a centrifuge to separate the protein parts of the phage, called the phage ghosts, from the rest of the infected cell. The scientists discovered that the labeled DNA appeared in the host cell, but not the phage ghosts, while the labeled proteins appeared in the phage ghosts, but not the host cell. In addition, the progeny of the T2 with labeled DNA were radioactive, while the progeny of the T2 with labeled proteins were not radioactive. Because genetic material is passed on from parent to offspring, Hershey and Chase concluded that DNA had to be the genetic material of T2. Their results helped convince the scientific community that DNA was the hereditary material.
So even with all of the other experiments, Hershey and Chase still had to do their experiment to prove, again, it's really not protein, it's DNA, we promise. So again, they used E. coli, they used a bacteriophage and they used radioisotopes to label either the DNA or the protein, and then they transformed that into E. coli, and they're like, oh, look, what do you know? It's DNA. Here's another visual of that. The protein coat, right? It infects the phage, it infects the bacteria. So they radio-labeled the DNA, they radio-labeled the protein, and then they waited to see which one infected the E. coli. And there you go. It was DNA. So just in case you weren't convinced before, it really is DNA. Here is a different demonstration of that, a summary of Hershey and Chase's experiment, and sure enough, it's DNA. It really is. Okay, transfection experiments were the same thing. E. coli treated with an enzyme lysosome. A lysosome lyses the outside of the cell. It destroys the outer wall of the bacteria, the peptidoglycan. The outer, and then you can enzymatically treat the cells because then they're sort of like naked. And when those protoplasts that are naked are used, the virus does not need to be intact. And so they can initiate phage multiplication. Okay, the process of infection by these viral nucleic acids, again, proved conclusively that DNA alone is the genetic material. In case you were not convinced, it is in fact the genetic material. Some more evidence, because you're probably still not convinced, right? You still think it's protein. So there's just a ton of evidence pointing to DNA. We have protein is abundant in cytoplasm, DNA is not. Mitochondria and chloroplasts perform genetic functions. DNA is present in those organelles. DNA is only found where primary genetic function is known to occur. Protein is found everywhere. It's all over the cell. DNA is not. So seriously, it's DNA. Just in case you didn't believe me, there's yet again another chart looking at the DNA content of haploid versus diploid cells in various species. And again, just proving to you, no, really, DNA is the genetic material. Some more evidence, in case you are still yet again unconvinced. Indirect evidence, mutagenesis. So when you use a UV light, it's mutagenic, right? Like, so this is just, let's not get the all-you-can-use tanning bed thing and be a weird orange color all year. No, because UV light, it's not great for your cells. So it causes mutagenesis. DNA and RNA absorb at 260 nanometers. Protein absorbs at 280 nanometers. So this means we can destroy different types of those cells to prove. So we can actually get rid of the DNA and RNA. And what do you know? The genetic material isn't happening because there's no DNA and RNA. So this is at 280 nanometers, no significant mutagenic effects are observed because the genetic material is DNA. Here again is the spectrum of that for the UV light. Recombinant DNA technology, splicing together DNA sequences from different organisms. So we can do this. It weirds people out. It's actually not that weird. We will talk about that when we get into transforming things. But the human hormone insulin is also made in other things. And so that way we can use recombinant DNA to produce human insulin in other things. Genomics, a full set of DNA sequences in organisms. We have almost everything sequenced at this point. And you can compare genomes of individuals to actually look at heritable disorders. All in the, what is it? DNA, not the proteins. Okay, now, just when I convinced you that it was DNA, remember I just spent all that time convincing you it's DNA? Sometimes it's RNA. Sometimes. Because, you know what? RNA is weird. Viruses are weird. And it's like a marriage made in heaven, right? RNA replicase is the enzyme needed for replication. Retroviruses, which are RNA-containing viruses, serve as templates in reverse transcriptase. So that means it goes from RNA to DNA. It's reversing the transcriptase. And that uses reverse transcription, happens with reverse transcriptase, and we can go from RNA backwards to DNA. And that's how we know that some RNA is the genetic material for viruses. Reverse transcriptase looks at RNA-dependent DNA polymerase enzymes, and one of those examples is HIV. It is a virus where RNA is the genetic material.